Hello my dear students, how are you all doing? Here we are with the next set of video where we are going to discuss the 2023 NEET question paper. I hope you remember that in the last set of video we have discussed that how exactly we can score 340 plus in biology, and 340 plus in biology is must to score at least 600 marks in your NEET examination. To become a good doctor, 630, 640 or more than that should be your target, isn't it? So here in this video, we'll be discussing the 2023 question paper. Why this question paper we are discussing? Why exactly 2023 paper itself right now? In this video, I'll be telling you that how you can approach for a particular question. How easily can you eliminate the one which is not correct, and how quickly you can come to a correct conclusion, right? So let's start. Hi, I'm Suparna, a biology faculty with Diksha, and I'll be here to tell you today about the first part of our lecture videos of solving 2023 paper, where I'm going to concentrate question number 101 to 135. This is the botany part of the question. Now, just for your information, I want to tell you that biology question paper will be starting mostly from 100 question. The question number will be 101 from where we will be starting forward. Here, biology is divided into two parts, zoology and botany. In the botany section, there is section A and section B, and same in zoology as well. So that means botany you will get section A, which is a compulsory part, and section B, there are options to choose. You will be getting 15 questions in section B, and out of that you can answer any 10. Now what if you are answering more than 10? Is it a problem? Generally, the trend is, what I suggest to you is, do not answer more than 10, because it has been observed that they consider while marking first 10 answers. And in that, if you do a mistake, obviously that is going to take a -1. And that's what I don't want, right? And I hope you also don't want that. So let's start with the section A of botany, which will be 101 to question number 135. In this part, we'll be talking about how you can solve the particular question. Remember, section A, all the questions are compulsory. That means if you are confident with the answer, you can obviously mark your OMR with the correct one, right? Let's get started. So the first question, what you can see. Okay, the question is quite big. Have patience to read the question. That is the first thing what I shall suggest. I know you will be running out of time. You'll be thinking that I have to hurry, I have to finish this, I have to answer this faster. But it's okay if you take a little more time to read the question and then come to a correct answer. Sometime that is really helpful. Let us read the question. As you can see, there is an instruction given. Let us read the instruction. Question number 101 says, given below are two statements. One labeled as assertion and assertion A, and the other labeled as reason R. Assertion A: the first stage of gametophyte in the life cycle of moss is a protonema stage. Reason or R is protonema develops directly from spores produced in capsule. And the option given over here, what is the question now? In the light of the above statements, choose the most appropriate answer from the questions below. And here we have the options as both A and R are correct, and R is the correct explanation of A. That means reason is the correct explanation of assertion. Option B, both A and R are correct, but R is not the correct explanation of A. Let us see the option 3, which says A is correct, but R is not correct or incorrect. And 4 is A is not correct and R is correct. So now these type of questions confuses us a lot. Statement-based questions makes a child very nervous because the question length is so big and you feel like, oh my God, I have to read. But see, if you observe, this question has been asked from, can you tell me which chapter? Yes, I hope you guessed it right. This is the question from your 11th grade Plant Kingdom chapter. And in Plant Kingdom chapter, these two sentences are exactly taken from NCERT. Let us read again. The first stage of gametophyte in the life cycle of moss is protonema stage. That is a correct statement. And protonema develops directly from spores produced in the capsule. And this is also exactly right, and this is the reason for assertion. That's why what will be the correct answer? Yes, you guessed it right. Both A and R are correct, and R is the correct explanation of A. And so that's why we are going to lock the answer as answer option 1. Right? I hope you got it. Let's move to the next question. The next question says, okay, small, pretty interesting. Let's see. In angiosperms, the haploid, diploid, and triploid structures of a fertilized embryo sac sequentially are. Now, what is the catch over here? You know that there is 3 things asked over there. What are the 3 things asked over here? One is haploid, diploid, and triploid. And one more thing over here is they ask sequentially. That means you have to say N, 2N, and 3N in a sequence where you can see this. N, 2N, and 3N where you can observe this. Got it? So let us see what are synergids. N. What is primary endosperm nucleus? It is 3N because after the fertilization. And what is zygote? It's obviously 2N because N plus N, a male gamete and a female nuclei is getting combined, and then we are getting a zygote which is 2N. So now is it in a sequence? What do you have to look for? N, 2N, and 3N. Antipodal cells, N. Synergids, N. Primary endosperm nucleus, you know it is 3N. So these 2 options are obviously not the correct answer. Let us see the third one. Synergids, N. Zygote, 2N. Primary endosperm nucleus, 3N. So till now I feel we have got the correct answer as option 3. Fourth one, synergids, antipodals, polar nuclei, all of these are N. So obviously this is also a wrong answer. So for this second question 102, the correct option is option 3 in a sequence. N, 2N, and 3N, they are arranged, and the answer is synergid, zygote, and PEN or primary endosperm nucleus. Correct? Let's go for seeing the next question. The next question, pretty easy question. You should not take more than 10 seconds to answer. I'll read out the question for you once. Movement and accumulation of ions across a membrane against their concentration gradient can be explained by. The catch over here, I know by now you would have already guessed the answer. Here, what is the point over here? Against concentration gradient. Now, when you want to move anything against the concentration gradient, what you need energy, right? And here, energy will come in the form of ATP. Wherever there is a requirement of ATP, we call that process as active transport. So that as easy as that. So the correct answer is active transport. It's not osmosis. Obviously, it is not facilitated diffusion, and passive transport, it's not possible at all. So what is the correct answer? Against concentration gradient, correct answer will be active transport. I hope you got this. Let's go to the next. Okay, there's an interesting one. Large, colorful, fragrant flowers with nectar are seen in. Now, why the flower will become very large, why it will become colorful, and why it will have nectar and fragrance? To attract the insects, birds, or something like that. Okay, birds will be different category, but here we are talking about large, colorful, and fragrant flowers which with nectar will be seen in those which is insect pollinated flower, not bat, not wind, or not bird pollinated. The color will be bright, so the insects will be attracted towards the flower, and that is why they will be insect pollinated, right? Let's go. All these are first year concept again. Okay, the phenomenon of pleiotrophism refers to, we move to the second year concepts now. What is pleiotrophism means over here? The phenomenon of pleiotrophism refers to, what is pleiotrophism? It is single gene carrying many characters. The best example if I wanted to give you is PKU, or which is called as phenylketonuria. Because of one defective gene, there are multiple characters of that particular person who will be disturbed, or they will show the disorder. So the correct answer for this will be one gene carrying many characters. So by the definition itself, by the question itself, you should be knowing, the definition should click in your mind. Which chapter it is? This is Mendelian law. That means principles of inheritance and variation from your standard 12th. Let's see what are the options given. Presence of several alleles of a single gene controlling a single crossover. No, I've told many. Then, presence of two alleles, each of two genes controlling a single trait. No, that is also not correct. Now, third option, a single gene affecting multiple phenotypic expression. This looks correct because what is the definition of pleiotropism? A single gene controlling many characters. So, a single gene affecting multiple phenotypic expression is the correct answer. Let us read the fourth option as well. More than two genes affecting single character. No, this is not the correct answer. So, what is our 105 correct answer? That correct answer is option 3. Okay, which hormone promotes internode or petiole elongation in deep water rice? This is question number 6. You have option as GA3, chitin, ethylene and 2,4-D. The correct answer over here is again option 3, ethylene. Ethylene helps in the ethylene or ethyl in the plant growth and development chapter. This hormone helps in the internode developments and petiole elongation in plants. I hope you got this, why it is ethylene GA3, which is helping in deliscence, and kinetin, it is promoting the cell division process in plant, and 2,4-D actually working as a weedicide or pesticide. So none of the three answers are correct, and that's why we chosen option 3. Let us go to the next question, which is 107. This is again second year chapter. So can you see the question pattern over here? One chapter is coming from your 11th standard and one another chapter is from 12th. You will be having mixed concepts also. I'll tell you where. Here the question is, among the evil quadret, which one is considered the most important cause driving extinction of species? Where you see extinction of species is taking place. The 4 options are given, which is evil quadret as per your second year or 12th grade knowledge. Habitat loss and fragmentation, overexploitation for economic gain, alien species invasions, and co-extinctions. Now here the question, it clearly says cause, what is the most important cause driving extinction of species? Why there will be an extinction of species? What is the important cause over here? See, if at all I say co-extinction, 2 species are getting extinct, but that is not the most important one. I hope you got it right. This alien species invention is when alien species comes in a particular area or in a particular water surface, that is alien species invention. Overexploitation for economic gain is not the most important cause, but if I consider habitat loss and fragmentation, that means when there is a habitat in a particular area, there is a large amount of loss because of any natural calamities or any other reason, then that causes a mass extinction or a large extinction of a important species. And if those species are endemic, then that is a loss for biodiversity, a huge loss of biodiversity, right? So what is supposed to be the correct answer over here? Habitat loss and fragmentation. Again, my dear student, please read the question properly and try to understand what exactly is expected to answer. Sometimes you will be, in between 2 options juggling, like, okay, is this the one, is this the one? At that point of time, take a second pause, read the question once more and try to understand what exactly is asked. You may get these questions again in the next year, who knows? But still, if you practice more and more papers, you will be obviously confident with what should be the correct answer for that, right? Let's go to the next slide to see the question number 108. Okay, this is a straightforward, easy question from biotechnology chapters. Upon exposure to UV radiation, DNA stained with ethidium bromide will show. You know this. Again, this is bright orange color. Remaining options are given just to, give or confuse you. This is a correct statement. Ethidium bromide, when a DNA is stained, it is going to appear bright orange under UV radiation. This statement you can just, input in your mind that ethidium bromide will give bright orange color to the DNA. Remaining options given as bright red, bright blue, bright yellow. Obviously, these are wrong answers. What is the correct answer then? It is bright orange color. So going with 108 question, option number 4. Question number 9. This is from the chapter of 11th standard botany part, which says that which micronutrient is required for splitting of water molecule during photosynthesis. So directly the question is from photosynthesis chapter. But here what you need to know about all these metals, that what is their function, and you should be thinking quick also. Got it, right? That means you have to think quick to come to the conclusion for this particular answer. Now here we have manganese, molybdenum, again magnesium, which is to confuse you. See, manganese and magnesium both are given. Sometimes you feel like, okay, it looks similar. And the fourth option is copper. So copper is helping a plant for its overall production. Okay, so what is the function of copper? It helps in overall metabolism. Okay, now what you have to look forward for the photosynthesis one. If you see magnesium is helping in some of the photosynthesis and respiration process, but it is not helping in splitting the water. It is helping in photosynthesis and it is also helping in respiration, but directly splitting of water is not done by magnesium. Molybdenum, on the other hand, is used for nitrogen metabolism. So obviously molybdenum is not a correct answer. Nitrogen metabolism. Now let us go back to the question to check what is the option asked. Which micronutrient is required for splitting of water? And among all these, the micronutrient which is required for splitting of water is option number 1, that is manganese. Manganese plays a very important role to liberate water during photosynthesis by splitting water molecule, and that is why we can say manganese is the correct answer over here. So here we are going to go with 109 option 1. 110th question says, here it is, axial placentation is observed in. This question is example based. That means you should be really thorough with plant anatomy and physiology in such a way that you should be able to answer the questions asked in this thing. So what is placentation? The arrangement of the ovule in the particular ovary of the plant. Now when we talk about the placentation, how many types of placentation are observed? Can you name them? Okay, let us name them. That is parietal, axial as already is given, free central, we can see free central, and we also can see marginal, right? So there are 4 types of placentation is observed. Here specifically they are asking for axial placentation. What do you mean by axial placentation? You will see the placentation, if you see the placentation, and let us see, it's like this, and you will see the presence of the ovule like in this pattern, right? Now this is the pattern of axial placentation. There are another 3 more. You will be seeing the marginal one, you will be seeing the parietal one, you will also be seeing the free central one. Now let us divide these examples quickly in a placentation wise. So what are the placentation we have? We have axial placentation, I'll write as A over here. We have parietal placentation, I'm going to write P over here, and then we have another 2 more, free central and marginal. So we have free central and free central and marginal. Okay, so these are the 4 type of placentation. Our question is to find out which of the plants are showing in the example as axial placentation only. So mustard, and you will be seeing there are how many examples are there? Let's count: mustard, cucumber and primrose. So in this, mustard is showing the parietal. So here I will be writing as mustard. Okay, then you have cucumber and primrose. Cucumber and also is going to show with mustard in the same way as parietal. So this is cucumber. So now see, we can easily eliminate option 1 from here because these 2 are not showing axial placentation. Now tell me quickly, which plant can show axial placentation out of this? China rose. Yes, China rose shows axial placentation. So now, without wasting more time, let us see what are the other options are available with China rose. China rose ke saath it is given as beans and lupin, which is beans, as you know, beans is like a bean and it has beads like this, which is obviously not a axial placentation, but rather it is a marginal placentation. So beans This is wrong. ये तो गलत है. So we cannot take this option. China rose, though it is showing exile placentation, but beans again over here is not. So this option is also cancelled for us. So then where we can see China rose once more in option number 4. So China rose, petunia and lemon. Yes, all these 3 are showing the exile placentation. So what are we going to lock with? China rose, petunia and lemon. Now you may ask, ma'am, why we left out with this tomato and pea and okay, tomato and China rose, tomato, petunia and lemon. All these 4 are actually showing exile placentation. So I can say tomato is there with exile placentation, but with this there is a P option, and P, it clearly says it is not exile placentation. So here only the options get cancelled, right? So this here we are going to go with 110th answer is option number 4. Again I am telling, be careful, have a quick thinking, remember what exactly the placentation is and how many types of placentations are possible. And if you remember one and find out what are the other examples, quickly remember what exactly in NCERT you would have studied. And I'll tell you, my dear students, none of these examples are from out of NCERT. All these options are given in NCERT. If you have forgotten, go and quickly check your first year textbook, the 11th grade. Okay, now this is an easy one, cell cycle and cell division. The process of appearance of recombination nodules occur of which substage of prophase 1 of meiosis. So they are asking over here in prophase 1 of meiosis 1. Prophase 1 of meiosis 1 are having how many steps? There are 5. What are those? Can you name? Leptotin, zygotin, pakitin, diplotin, diakinesis. I say it as LZPDD. To my student, I always teach them like this. LZPDD. Leptotin, zygotin, pakitin, diplotin and diakinesis. Now, the question over here is appearance of recombination nodules. We are going to talk about appearance of recombination nodule. At this point, the chromosomes are going to come together and they are going to form nodule formation or looping with each other. And this is obviously seen in the 3rd stage of the meiosis, prophase 1 of meiosis, that is in the pakitin stage. So here the correct answer is pakitin. In the zygotin, they are just forming the bivalent stage, they are forming the tetrad, but the nodule formation has not taken place in zygotin. Leptotin obviously is not option, is not there at all. And diplotin and diakinesis, I hope you are very clear with it. What happens in diplotin? Diplotin mein we see the X. What is that X over here? It is the chiasmatic formation, right? And diakinesis mein by the time the almost chromosomes would have left each other and they're going to start coming to the middle part or the mid plate of the cell, and they're going to get ready for the cell division. I mean, they're going to get ready for pulling up. I think one more question, I think I've seen it. Let us see those questions and I think we can discuss a little more on that. Is this clear? The stage where we see the recombination nodule in prophase 1 is zygotin. I mean, not sorry, zygotin, it's a pakitin. So we are going to lock with option 2, pakitin. Right, let us go to the next stage question. Oh, again the direct question, you see. The reaction center in PS2 has an absorption maxima at, quickly, it is 680 nanometer. What are the other options? 700. 700 is for PS1. 1 is always big. 1 is 700. And what is for PS2? PS2 is 680 nanometer. Okay, so other 2 options doesn't make any sense. So we are not going to look into that. We'll go to the next one. Unequivocal proof of DNA, proof that DNA is a genetic material, was first proposed by. Which are the scientists propose that DNA is a genetic material without any doubt? For this, if you quickly revise, if you quickly can recall the scientists and their experiments explained in the chapter Molecular Basis of Inheritance. If you remember there, okay, Frederick Griffith experiment is for transforming principle. So Griffith, what he worked on? He worked on transforming principle. Alfred Hershey and Martha Chase. Hershey and Chase actually has done the experiment on, I hope you remember the Hershey-Chase experiment, which is very important with the bacteriophage and E. coli, where they have exposed the bacteriophage with radioactive phosphorus and the other one is radioactive sulfur, and they checked out where exactly the radioactivity is found, and finally they concluded that it is found in the genetic material. So they concluded actually the DNA is a genetic material. So this is supposed to be the correct answer as per the experiment done. Avery, MacLeod and McCarthy, if you remember these, they also worked on the transforming principle over here. They also checked with the transforming principle, right? And Wilkinson and Franklin, they exactly done the X-ray diffraction technology of DNA. They worked on X-ray diffraction technology to find out the structure of the DNA. So Wilkinson and Franklin works on X-ray diffraction technique. Frederick Griffith was working on the experiment of transforming principle, and we also have Avery, MacLeod and McCarthy working on the same to prove that what exactly the transforming principle is all about. So the correct answer for that the DNA is a genetic material is proposed and proven by, we cannot have any more doubt by seeing their experimentation. It was so good, and that is Alfred Hershey and Martha Chase's Hershey and Chase experiment, commonly called in our NCERT, and we read it in detail. Okay, we'll go to the next one. The next slide says, among eukaryotes, replication of DNA that takes place in. Now this is very easy question, I will say. This question is directly can be told as synthetic phase or S phase. In the cell cycle, a cell cycle, if you draw a cell cycle is divided into a part where there is a division going on, what we call it as M phase, or where we have divided with prophase, metaphase, anaphase, telophase and the karyokinesis part. So this particular part is M phase, and the remaining part is called as interphase. During the interphase again can be divided into three parts, G1, S and G2 phase. Now what happens in G1? Cell is preparing for doubling the DNA, and in the S phase actually the DNA replication will take place. The amount of DNA will be doubled, correct? G2 phase, they will do all the remaining synthesis, and then the cell will go for a cell division phase, starting with prophase, metaphase, anaphase, telophase and cytokinesis, right? Cytokinesis. So this is how the cell division takes place. Clearly it says S phase is the one where the DNA replication takes place. It's a very easy question, right? Let's move for the next question. Question number 115. Question 115 says that in tissue culture experiments, leaf mesophyll cells are put in a culture medium, form callus. To form callus, they are forming the callus. This phenomenon may be called as, here the leaf mesophyll cells are actually de-differentiating to form the callus. This is the correct direct question answer, that is the process by which the callus formation is taking place by the leaf mesophyll cell is called as de-differentiation process. And do we have that option? Yes, is the second option, de-differentiation. Obviously it is not a differentiation because it is not the growth for the first time of the plant. It is we are taking the mesophyll cells and growing it biotechnologically to form the callus, and this is de-differentiation. Development and sentience, obviously these two are not the correct answer. So here we get the correct answer as option number 2, that is de-differentiation, right? Direct question. We'll move to the next. Cellulose does not form blue color with iodine because. Does cellulose disaccharide? No, right? What is this? What is cellulose? Come on, recall from biomolecule chapter. Cellulose is a, as soon as I start writing POLY, you will be telling that, ma'am, we remember it is a polysaccharide, right? Right. So cellulose is a polysaccharide. So option 1 is wrong. Cellulose, it is a helical molecule. It is not exactly, we can say it is a helical molecule, but it contains a complex helix structure. So see the sentence, what I'm telling is correctly matching with option number 3. Actually, it does not contain a complex helical structure, whether it is simple helical structure, and cannot hold the iodine molecule. It breaks down the iodine when it reacts with it. Obviously, is not the correct answer. So here 1, 2 and 4, all these 3 options are wrong options. So what we are going to go with? We are going to go with the third option. Cellulose does not form blue color with iodine. Why is it so? Because it does not contain a complex helix and hence cannot hold iodine molecule, and it is, cellulose is a polysaccharide over here, so it cannot be considered as disaccharide. Now, next question, we are going to go for 117. Okay, again, it is going to the first year 11th standard question. Spraying of which of the following phytohormone on juvenile conifers helps hastening the maturity period that leads early seed production? You need early seed production. That's what is the point over here. You need to go for a early seed production, and what you have to do, you need a phytohormone so that you can get a early seed production, right? So for this, we have first option as indole 3 butyric acid, option 2 gibberellic acid, option 3 zeatin, and option 4 as abscisic acid. I think by now you would have already guessed the answer. Still, let me say what exactly it is. Indole 3 butyric acid, it helps in promoting growth and development of the root, flower, and fruit. So it is helping the root, flower, and fruit development. It is not directly connected with seed production. Whereas gibberellic acid, it is helping in the maturity period for the seed to grow faster, and gibberellic acid is actually doing this process. So gibberellic acid is the correct answer, that is option 2. But I want to explain 3 and 4 as well to to be in a safer side, that okay, we are going with the correct option only. Option 3, zeatin, is a cytokinin. So cytokinin generally helps in self growth and cell division. So it is not cell growth and cell division directly, but we are going to have a maturity period, hastening the maturity period, and you are going to get an early seed production. So it is not because of cell division and cell growth. And abscisic acid is obviously not possible, because I hope you know that it is suppressing the growth. It's not allowing the plant to grow. And here we are talking about the growth. So obviously this cannot be the correct answer. So here we are going to go with gibberellic acid as a correct answer. Next question, let's go 118 question. Again, statement based question. Let's read the question first. Given below are two statements. Statement 1, the forces generated transpiration can lift a xylem-sized column of water over 130 meters of height. Statement 2 says that transpiration cools leaf surface sometime 10 to 15 degrees evaporative cooling. In the light of the above statement, choose the most appropriate answer from both the options given. Now here this is a statement, not assertion reasoning. There is a clear difference between assertion reasoning questions and statement based question. See, assertion reasoning question was the first question, if you remember, when we started solving. There will get two questions. One will be the correct answer and one will be the reason. Both of them may be correct. One could be the correct explanation of the first one. That was the pattern of assertion reasoning question. When we talk about the statement based question, here the options clearly says that either statement 1 is correct or statement 2 is correct, or the option can be both the statement 1 and 2 are correct. So here what is the question given? The forces generated transpiration can lift xylem-sized column of water over 130 meters of height. Can the force of transpiration go to 130 meters of height or not? Yes, it is possible by the transpiration pull the water by the xylem tissues up to 130 meters of height. You know that there are big giant plants, and they by the help of transpiration pull itself, the water can go. So statement 1 is the right answer. Right option. Let us see, transpiration cools leaf surface sometime 10 to 15 degrees evaporate cooling. Yes, leaf surface can be cooled by the transpiration process, and statement 2 is also correct. So statement 1 and statement 2 both are correct. Let's read the option now. Both statement 1 and statement 2 are correct. Okay, here we go. We have the answer with us. Second option says both statement 1 and statement 2 are incorrect. Third option is statement 1 is correct, but statement 2 is incorrect. Fourth option is statement 1 is incorrect and statement 2 is correct. So first, when you are doing this statement based questions, take a pause, read the question before looking at to the optional key, because you know statement based questions are like this type only, that you will be getting two options. One is both correct, another will get both incorrect. Third option will be one correct, other incorrect, and vice versa. That means statement based questions will follow the same pattern in your term also when you will be writing your NEET exam. At that point of time, if you see statement 1 is correct, put a tick next to it. If you also see the statement 2 is correct, put a tick or put a cross, so that you will be very confident, because when you read the option, go back to the question, you will be completely confused. You are in an exam hall and there are many children sitting next to you, and you all are competing for that golden marks of 650 plus. And at that point of time, there is no chance of confusion or hesitation. Read it calmly. If you get the correct answer, just with a pencil, you just put a tick over there, so that you will be confident enough when you read the question, you can just come to the answers quickly. I hope this will help you. Let us see the next question, question number 119. Family Fabaceae differs from Solanaceae and Liliaceae. With respect to the statement, pick out the characteristics specific to family Fabaceae but not found in Solanaceae or Liliaceae. So you have to find out the specific character which you see in Fabaceae, but you're not finding it in other two. So here it is, the options given over here is diadelphous and diathecous anther, polyadelphous and epithecous stamen, monoadelphous and monothecous anthers, epiphyllous and diathecous anther. All these concepts are given with respect to these three families, Fabaceae, Solanaceae and Liliaceae. Now what you have to find it out, that which one is a specific characteristic in Fabaceae but we don't find it in Solanaceae and Liliaceae. Now in Solanaceae you find this one, polydelphous and epipetalous, and the anther over here is diathecous anther. So here they also have a diathecous anther in this second option I'm talking about, okay? In that with respect to the family Solanaceae. What exactly for family Liliaceae? Liliaceae is having polyandrous. Liliaceae is having polyandrous. The option is not given over here. Anyway, we're not looking for Liliaceae also. Liliaceae is polyandrous and it is epiphyllous. So these two also are specific character of Liliaceae. Now Fabaceae shows unique characteristics by, I'll erase this so that it becomes very clear for us. So what is the question again? The family Fabaceae, we see the character, but we do not see any of these characters matching with Solanaceae or Liliaceae. And the correct answer over here is this one, diadelphous and dioecious anther, which is uniquely seen in Fabaceae, which pick out the characteristics which is specifying the family Fabaceae. Right? So correct option over here is option 1. Let's go to the next one. Express sequence tags or ESTs refers to. This comes from our human genome project, where the approach was, how can we find out the genomic structure? There are two approaches. One is EST, another one is SA. SA is sequence annotation. What exactly is done in EST? There, what are the genes which are getting expressed in terms of RNA, mRNA, that has to be read and that need to be marked, or that need to be, we are going to find out the nucleotide of those. That was the express sequence tags motto. So here, all genes that are expressed as RNA, yes, this is actually the correct answer. But I want to read all the other options also. All genes that are expressed as proteins, not exactly the proteins, but we are talking about with respect to RNA. So this all genes that are expressed as protein is not the correct answer. All genes, whether expressed or unexpressed. This was actually approach of SA, sequence annotation, right? Sequence annotation means we're going to go for sequence annotating no matter whatever it is it expressed or not expressed. We are not bothered about that. We're going to read all or label all the nucleotide present in that particular chromosome. So here certain important expressed gene is obviously not the answer. So here what is the correct answer? All genes that are expressed as RNA. Remember here, whatever genes is expressed as RNA may not be always forming the protein. There can be other function of those RNAs also. And that is why if you would have chosen the answer as protein, you would have been going wrong. Express sequence tag, they followed the technique where they found out what are the RNAs that is getting formed from the chromosome or from the DNA. So that is what is ESTs, right? So correct option, you should be a little careful when RNA and protein is coming over here. The correct option is option 1, which is as a RNA. Next one, question number 121. Identify the correct statement. Here you have to be again careful because you have options. If you see directly option A, B, C only, B, C, D only, C, D, E only, D, E, A only. Now they are asking you to choose what they are choosing, correct statement. What I told you what to do for statement questions, you just put a tick if it is a correct answer. Detrivores perform fragmentation. Is it the correct statement? Yes, retrievers do perform fragmentation. So here I'm going to put a tick over here. This is the correct option. Let's see the next one. The humus is further degraded by some microbes during mineralization. This is also a correct answer. So A and B. Now at this point, you can do one more thing. You can do one more thing in terms like, see where A and B both options are given, you can choose that directly because both of these are correct. So out of these options where you have A and B together, that should be the correct one. Do we have multiple options with A, B? No. See, A, B, C only, B, C, D only, C, D, E only. Where you find A and B both are given in the first one itself. So without reading the third statement, you can directly go to answer as option 1 because both the statement what you find as correct. So if you're lucky enough, you may get first two statement correct and the options also doesn't have a repeated things, right? So you can see A, B is only seen in this question number 1. So to be in safer side, I'm going to read and I'm going to explain it. But in exam, when you want to save time, my dear children, follow this rule. If at all you see the options and with the options, the statements are matching, at least in this case, 75% or plus is matching with the options, then go for it because this is the correct answer. C says, water-soluble inorganic nutrients go down into the soil and get precipitated by a process called leaching. This is also a correct statement. So see here, A, B, C only is correct. That means D and E is supposed to be a wrong statement. Let's read. The detritus food chain begins with living organism. Obviously not. You know that, right? Detritus food chain starts with the dead and decaying organism. So this is not a correct answer. Earthworms break down detritus into smaller particles by a process called catabolism. No, this is also not a correct answer. Okay, I like these two options. Anyway, D and E is wrong. So A, B, C is correct and we have already chosen it. So you have saved some time over here while answering your paper, right? 121 is done. We'll go to the next. Okay, this is nice. The thickness of ozone in a column of an air in the atmosphere is measured in terms of, okay, it's very easy. It is correct answer is Dobson unit. We have discussed this. It is there in the environmental issues chapter. I think this year you do not have environmental issues as in the syllabus, but yeah, you can obviously have some extended knowledge. The thickness of the ozone layer, how much ozone layer is there in the ozonosphere, is measured by a unit, and that unit is called as Dobson unit. There is one thing here also given as decibels. I hope you know what is decibel. What is decibel is used for to find out the noise, intensity of the noise. Decameter, okay, it's very easy. This is to measure length, meter and decameter. Kilobase, if you want to know what is kilobase, kilobase is actually used to find out the length of DNA or RNA. You can put a star and write it in your book. This is to find out the length of DNA or RNA, right? So this is FYI for your information that you can always use this. What is kilobase? Kilobase is to find out the length of DNA or RNA. Got it? So anyway, we got the correct answer as Dobson unit. This is the environmental issues chapter. Don't worry, if it is removed, these questions will not be coming. Let's go to the next question, question number 123. Again, statement-based question. Given below are two statements. One is labeled as assertion. Okay, this assertion reasoning question. A and the other is labeled as reason R. Assertion says that late toad has fewer xylary element with narrow vessels. Reason is cambium is less active in winters. Now let us see the options. In the light of the above statements, see both the statements are correct. First thing is you have to find out that if the statements are correct or not. What I told you, see late wood has fewer xylary experiment and narrow vessels. This is a correct statement. And cambium is less active in winter. This is also a correct statement. So both these things are correct. Now let us see in the light of this above statement, we have to choose the correct answer. Let's put some light onto it and find it out. Both A and R are true. And R is the correct explanation of A. Oh my God, the first answer itself is the correct answer. I got my answer over here. Okay, both A and R are true, but R is not the correct explanation. No, obviously not. A is true, but R is false. Wrong, wrong, wrong. A is false, but R is true. Wrong. So we got the answer. Both A and R are true, and R is the correct explanation of A. So here I'll see this type of questions are actually considered to be easy questions. You know, you think that, oh my God, there is so much to read. But see, when you have a direct approach to a question, those questions will be considered as easy question. So both A and R are true, and R is the correct explanation of A. In winter, the cambium is less active, and they forms very few xylary element, and which is this wood is called as autumn wood, or it is called as or late wood, which is called as autumn wood or late wood. So obviously both the statements, again NCERT pakka line leke they are put over here. Read NCERT, okay, don't ignore. Okay, this is again a easy question. Which of the following stages of meiosis involves division of centromere? Division of centromere. Which of the following stages of meiosis involves in division of centromere? Okay, quick revision. Meiosis is divided into meiosis 1. Let me not write like meiosis M1, but let me write it like meiosis 1 and meiosis 2, right? So meiosis is divided into meiosis 1 and meiosis 2, and they are asking the division of the centromere. Centromere division will be only taking place during the anaphase stage, no matter what. It is not in the metaphase. Metaphase state may, what happens to the cells? They get arranged to the equatorial plane. The chromosomes get arranged in the equatorial plate region, right? So metaphase 1 and metaphase 2, not possible to be a correct answer of error. Both the options are wrong options. And whenever it comes with the division of the centromere, you should know that that is anaphase. What happens in the telophase state? You know that, right? In telophase state, the chromosomes would have been pulled towards the pole. Chromosomes would have been pulled towards the pole, right? So that means the centromere would have been already been divided over here. We do not have the centromere in the division state. The following stage of meiosis involves division of the centromere. Correct answer is anaphase 2, right? So this is direct. You should hardly need 10 seconds to answer these type of questions. Question number 125. The historic convention on biological diversity, the Earth Summit, was held in Rio de Janeiro in the year. This question can come in two different ways. When the summit was held, that is in Rio de Janeiro. This question also can come as when as the Earth Summit was, what is the pledge they took to complete the whole plan, whatever they had in Earth Summit. That is 2002. But in 1992, this is this Rio de Janeiro Earth Summit that was taken place at that in the year 1992. It is a direct question, 1 marker question, so you should be knowing this answer easily. We'll do a little bit calculation over here. How many ATP and NADPH2 are required for the synthesis of one molecule of glucose during Kelvin cycle? Quick way of understanding. Remember that one molecule of carbon dioxide will be giving 3 molecules of ATP in Kelvin cycle. 3 molecules of ATP in a Kelvin cycle. You can just write it down somewhere for easy calculation, and it will be forming 2 NADPH2. Okay? So what exactly it is? One molecule of carbon dioxide is giving you 3 molecules of ATP and 2 NADPH. I mean, it is needed. 1 molecule of carbon dioxide needs 3 molecule of ATP and 2 molecule of NADPH2, right? For the Kelvin cycle. Now the question is, how many ATP and NADPH2 are required? You require to synthesize glucose. How many carbons are there in glucose, my dear children? You know that glucose is a hexose, so it is C6H12O6. How many you have? 1 carbon over here. To make glucose, as the Kelvin cycle says, how many carbon dioxide you need? You need 6 CO2. So what do you do? You have to do a simple calculation of multiplying this number with 6 and this number also with 6 to get 12 ATP and 18 ATP. Where is the eraser? Here is the eraser. So we will write it. You need 18 ATP and 12 NADPH2, right? So what is the correct answer? 18 ATP, 12 NADPH2. 18 ATP, 12 NADPH2. Correct? So this is the correct answer. So you have to remember, to produce one carbon dioxide molecule requires 3 ATP, 2 NADPH2. 1, 3, 2. 1 ATP, 1 carbon dioxide, 3 ATP, 2 NADPH2 is required. You need to produce 6 carbon compound glucose. So multiply with 6. You get your answer as 18 and 12. Done? Let's go for the next. In the equation, the question is in the equation, GPP minus R is equal to NPP. Okay, here they're good. I like it here because they've given the full form of GPP and NPP. You may not get this in your paper because you're supposed to know these are all NCERT terms. So GPP, they have told what is the GPP meaning over here. It's a gross primary productivity. NPP is net primary productivity. What is R? R is actually the loss. It is the respiratory loss. It is again a direct question. Less than 10 seconds, I need to answer this. That should be your confidence. R is respiratory loss, the amount of energy lost during respiration by the plants, that is respiratory loss. So in this one, it is not photosynthetic active radiation, it is not respiratory quotient, which feels like respiratory quotient, but this respiratory quotient and respiratory loss are two different things. We are talking with respect to gross primary productivity. Reproductive allocation, no, that's the wrong answer. So in these type of questions, I want you to have a direct approach. We are going to practice more and more papers. With this video session, we'll be practicing many questions like this. We'll be practicing smaller questions. We'll be specifically doing the chapter specific question answers. We'll be doing all the assertion reasoning questions once, all the statement based questions once. So once you are exposed with many questions in a different way, which can be asked, you will be so confident to write the exam that as soon as you see the question, you'll be like, oh, I know this, I've done this. Read it. If you find it is matching with your previous questions, what you have practiced, you can just mark the answers. So that is what is our whole effort is to get you that confident, to give you that confidence that I know this answer, I'm going to do it, right? Chalo, we'll go for the next one. Question number 128. Okay, chota sa question hai, small one. During the purification process for recombinant DNA technology, addition of chilled ethanol precipitate out. Oh my God, again, this is a very easy question. It precipitate out DNA. Yes, we are talking about the DNA technology. The clue over here, we are talking about recombinant DNA technology. And what it is precipitating out? It cannot precipitate proteins. Histone is a protein. It is not precipitating protein, obviously, no. Histone is a protein. Polysaccharides, no. It is not segregating polysaccharides because polysaccharides saccharide like carbohydrate, no. And RNA, no, because we are recombinant DNA technology. We are working with recombinant DNA technology. And here, what is going to be precipitated out? Correct answer is DNA. Yes, you guessed it correct. Sometimes the questions will be simple like this also. Next, question number 129. Let's go for 129. What is the role of RNA polymerase III in the process of transcription in eukaryote? There is a clear division of labor in eukaryotic organism when it comes with respect to RNA polymerase. As you know, RNA polymerase 1, 2 and 3 is present in a eukaryotic cell, unlike prokaryotic cell. Correct now? So now what is the question over here? You have to find out what is it RNA polymerase III doing. RNA polymerase III basically transcribes the snRNA, rRNA and tRNA. So, that is what is the correct answer. So, transcription of tRNA, 5S, rRNA and snRNA, these three are produced by it is like, it is RNA polymerase III is required for these processing. You have polymerase 1 for 5.8S RNA and 18S and 28S RNA, and you have polymerase 2 as hnRNA or the processor RNA, and RNA polymerase 3, which is a question, sTRNA, 5S rRNA and snRNA. Okay, so this question is also again a direct and quick to be answered. These questions are easy going. 10 seconds, as I'm telling, to answer these. 10 to, okay, you take 30 seconds max to max. But within that, your idea should be so clear, crystal clear, that you can immediately answer this. Let's go to the next one. Where is it? Question number 130. What is the function of tassels in the corn cob? Okay, corn cob, the corn. The tassels, that feather-like extensions are there. What is its function? What does it do? It helps to trap the pollen grain No. It is not to disperse the pollen grain. Don't get confused. Tassels are not to disperse. It is to trap, right? When the pollen grains are coming near to it, it's to trap. It is not to attract insect or not to protect the seed. These tassels are there in the corn cob. The corn, you know, right? The corn. In that you find that hairy type of appearance. Those are the tassels of the corn cob. It is there to trap the pollen grain, right? Let's go to the next question. Question number 131 is saying that identify the pair of heteroporous pteridophytes among the following. Can you tell me what is the meaning of heteroporous? Hetero, two different type of spores are produced, then that is called as heteroporous. Okay, so heteroporous pair of pteridophytes among all the following. So there can be one more type. What are those? Those are called as homosporous. That is called as homosporous. Homosporous means both the gametes will be similar, both the spores will be similar. And on this context, if you remember, you have coleotom, then lycopodium, and you have equistem. Those are homoporous. So now, can you see? If you identify the question in a little different way, if you think in another way, if you can remember this homoporous, then we are left up with one option. Did you get it? You omitted all the wrong options and your board itself says, then what is the heteroporous one? As we know, Saligenilia and Salvinia are heterosporous. So what I did, I just found out how many homosporous were. That was easy for me to understand. You can remember it anyway. Homosporous, I could remember easily the examples as Lycopodium, coleotom and equistem. These three were homosporous. So as we see, these three options are here in all the three options. And my question is to find heterosporous pteridophytes. What I left up with is Saligenilia and Salvinia. So we are going to log this answer as option 2. Next question chalte hain. 132. Okay. Again, a very straightforward question. Just directly NCERT line and very important concepts. The question is, in gene gun method, in gene gun method to introduce alien DNA into the host cell, microparticles of dash metal are used. Dash metals are used. So, okay, can you see here? They have also given a clue over here. They have used dash metal are used. So, I mean, there can be more options, but here the correct option is tungsten or gold. So the correct option is option 3. Copper, zinc and silver is not used in gene gun when you directly send the alien DNA into the host cell by gene gun technique, where you use the microparticles which will be having tungsten and gold on it, not the other one. Again, direct questions from NCERT, easy to answer. I will say botany was comparatively easy as per it, what I'm analyzing right now. Looks like most of the question, we need not to think so much, think so critically. Oh my God, can I, do I have to do a lot of calculations? Do I have to think so much? No, it looks very comparatively easy to me. Okay, again, statement 1 and statement 2. And you have to find out to choose the correct answer. Endark and exarch are the terms often used for describing the position of secondary xylem in the plant body. Is it the correct answer? What I told you first what do you do for the first time when you read the question? You see this and think that is it correct or not? No. Now at this point of time, option 1 is incorrect. Okay, statement 1 is incorrect. So I think I'm heading towards this because, okay, that's going to be 2 also. They're telling statement 1 and 2 both are false. Let's see, read this one. Exarch condition is the most common feature of the root system. This is a true statement. So this is true and this is wrong. Now tell me, my dear children, what would be the correct answer? It's very obvious. Option 4. Statement 1 is incorrect. Statement 2 is true. That means we are going to eliminate as soon as we read. As you read, you are left with 2 options, either 2 or 3. So immediately you read the second statement and you got to come to the conclusion that this is how you have to answer, right? Question number 134. Frequency of recombination between gene pairs on the same chromosome as a measure of distance between genes to map their position on chromosome. was used for the first time by? This is from the chapter Principles of Inheritance and Variation, where we discussed Mendel laws in the initial part of the chapter, and then we went to linkage and recombination. Here they are using the concept of linkage and recombination, see frequency of recombination between the gene pairs. What exactly will be for the frequency of the gene pair? For that, which scientist has told, or which scientist has done the gene mapping? In Human Genome Project also, that particular scientist's work is used in a large amount, and we really appreciate that the way he has done the gene mapping. It is not Thomas Hunt Morgan or not Sutton and Boveri. This is the this work is done by Alfred Stuart Burnett. He was actually working under Thomas Hunt Morgan when the experimentation was going on, and he found out the gene mapping technique at that time. He exactly mapped the gene, what exactly the experiment was going with the Drosophila, and that technique later was used in Human Genome Project, right? So what I mean to say over here is, if you have read the chapter confidently, you could have concluded this, not Henking, not Thomas and Morgan, or Sutton and Boveri. Alfred Stuart Burnett found out the frequency of recombination, where how the genes are linked together. Okay, Sutton and Boveri, they proposed the chromosomal theory of inheritance, and we got Henking, who discovered X chromosome. If you have forgotten, this is the same chapter concept. He discovered X chromosome, Hana? Chromosome, remembered? Yeah, Henking. Well, the sex determination part, we have this. We have spoken about Henking. And what about the Thomas Hunt Morgan? He actually proposed the chromosomal theory of inheritance. He has also a lot of contribution towards this. But the work which was done to map the gene was Alfred Stuart Burnett. So let's see the question. Next question, 135. Again, assertion statement based questions. Okay, no, assertion reasoning based question. Given below are the two statements. One is labeled as assertion A and the other is labeled as reason R. Assertion A. ATP is used at 2 steps in glycolysis. Okay, correct option. Reason R. First ATP is used in converting glucose into glucose 6-phosphate, and the second ATP is used to convert of glucose 6-phosphate into fructose 6-phosphate to fructose 1,6-diphosphate. Now you have to find out which answers are correct. See, ATP is used at 2 steps in glycolysis. This is a correct answer, correct step, right? We need ATP in both the steps. First ATP, what you are using is, you are starting glycolysis with glucose, right? So glucose, say what you are preparing, you are making glucose, chalo, it is written over there, 6-phosphate. What you are doing, in the 6th position of the glucose molecule, you are adding a phosphate group. So for that, obviously, you need ATP. So this is your ATP number 1 which is used. Whereas the second ATP is used? Second ATP is used to convert fructose 6-phosphate to fructose 1,6-diphosphate. Fructose 1,6-diphosphate. See here again one more thing, as you are using the word diphosphate, right? You are using the word diphosphate. That means what you are doing? You are taking 2 phosphate molecule. Diphosphate means what? You are having 2 phosphate molecule, right? So here also you need one more ATP. You need one more ATP over here. So this is the first ATP, and here you have the second ATP. So here 1 ATP, here 2 ATP. So first ATP is used for converting of glucose to glucose 6-phosphate, and second ATP is used for fructose 6-phosphate to fructose 1,6-diphosphate. Both of these are correct. So both A and R are true, and R is the correct explanation of A, because there are 2 glucose is used and we are getting 2 ATP for this process. Okay, so we'll see that what exactly we have. So we are actually done with all the 35 questions of as we planned for the first session in botany. Okay, next video, what we're going to do is, we'll be continuing solving these question paper of NEET 2023. In that, I'll be starting with the section B of botany, where you will be getting the optional questions, but obviously I'm not going to choose the options for you. I'll be doing all the 15 questions, and we also will be starting with the zoology part of it. Okay, so next set of videos which is going to come up, which where we'll be discussing the next part of the questions containing in the biology section, botany section, remaining botany section, and the zoology part of it, right? So this video sessions will be helping you a lot to understand a question, to approach a question and how to think quickly, in some cases eliminating the wrong answers. Remember, our main aim is to not to go for any negative marking, and for that we are going to try to answer all those questions as correct as possible, and we are not going to do any mistake over there, right? So we'll be having more sessions like this where we'll be discussing previous year question paper. We'll be talking chapter wise specific questions, and also I have a plan to make a video session where we can have a assertion reasoning based questions and statement based questions put together, at least 10 for that particular chapter or topic, and the probable questions what you may get. I think those will be helpful. I hope you like this session. Don't forget to subscribe and hit the like button. Thank you.